Hi guys, in this video we're going to be talking about a group of enzymes found in yeast called zymase, but I just thought I would start off with an introduction about what biocatalysts are in general. So biocatalysts literally are just as they say they are. They're biological catalysts. They're biological because they're found in biological systems, and they're catalysts because they speed up reactions. So this encompasses things such as enzymes, and cells themselves can actually act as biocatalysts. And there are thousands of biocatalysts in the human body and in all other living organisms. It's basically the way that we operate. So in this video we're going to talk about zymase. So zymase is a complex of 10 different enzymes responsible for the breakdown of glucose into CO2 and carbon dioxide, and in doing so they get energy. So the enzymes that make up zymase are hexokinase, phosphoglucose isomerase, phosphofructokinase, aldolase, triose phosphate isomerase, glyceraldehyde phosphate dehydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, and pyruvate kinase. So a bit of a mouthful. There's a lot of big names and a lot of biocatalysts involved in this reaction, and they work as a cycle. So the idea of biocatalysts is that they can actually be much more green alternatives to things like metal catalysts or not using catalysts in general. So if you use enzymes or cells as biocatalysts, you can imagine they can biodegrade, they're renewable, and also they can react under quite ambient conditions. They can react at room temperature and in water, and for those reasons they are seen as being a really good alternative, and it's something that's up and coming now. So hope you guys enjoy the video.
Most of us use biocatalysis around the home. Simply by baking homemade bread, you're using biocatalysis. Catalysis is important for reducing the energy consumption and the waste production in a chemical reaction. Yeast has served humans for thousands of years in baking bread and making beer and wine. It is able to actively convert sugars into ethanol and carbon dioxide through use of several enzymes. Enzymes are remarkable natural catalysts that are highly selective and reduce activation energy of a reaction. Yeast is mainly used for two processes: alcohol fermentation, in which the ethanol product is the most important, and baking dough, where carbon dioxide is the utilized product. The agent responsible for the fermenting activity of the extracted juice is a dissolved substance called zymase. The zymase reaction is an excellent example of design for degradation. Only a small amount of yeast is necessary to initiate the reaction, and the yeast reproduces while the reaction is occurring. Zymase was initially identified in yeast as the biochemical activity that was able to convert sugar into ethanol and carbon dioxide. Zymase activity was discovered long before biologists understood much about chemical reactions in the cell. And since then, the biochemical pathways that convert sugar into carbon dioxide and ethanol have been completely solved. The glycolysis pathway converts glucose into pyruvate. From there, pyruvate can be respired to carbon dioxide if oxygen is present, or fermented into ethanol under anaerobic conditions. Once the reaction has reached completion, the yeast dies, and the ethanol and all byproducts are easily, rapidly, and harmlessly degraded in the environment. This makes evident the green chemistry principles that apply to biocatalysis, particularly in the reaction of zymase.
Now we're talking about how zymase apply to green chemistry. There are two ways to produce ethanol. Firstly, hydration of ethene. We pass the steam of ethene and water to phosphoric acid with this catalyst. And the second method is fermentation of sugar with zymase. Now we are comparing pros and cons for both reactions. For the rate of reaction, hydration of ethene is faster than fermentation, because hydration of ethene uses high temperature and pressure, meaning that it requires much more energy input, but fermentation uses only normal temperature and pressure. Lastly, fermentation uses plant material, which is renewable, but hydration of ethene uses crude oil, which is non-renewable. So in this case, fermentation is better for the production of ethanol.
Hello, my name is Pakson, and I'm here to talk more about zymase, the enzyme within yeast. You see, yeast is a household product used for years and years and years. In fact, during the ancient Egyptian times, where we worship cats, it has been active for over 5,000 years. The ancient Egyptians used yeast in their bread to make ethanol and carbon dioxide to make the bread even more delicious. Don't you agree? But okay. So we have to say that there has not been a more greener chemistry than the enzyme zymase within yeast. Yeast has been used for years and years and years and have never been replaced by bare green chemistry. It's an absolutely correct pathway. You see, yeast is a fungus. It's a fungus that has great reproductive abilities. So it's highly renewable. There haven't been a more efficient industrial way for yeast to be replaced with.